So I will start. So today I'm going to talk about how we can predict metabolism to engineer the bioproduction of natural products, in particular those with interesting pharmaceutical properties. But first of all, here's a little bit the outline of the talk today. I will first give you an introduction on what we know today about metabolism, and then I will continue to show you the tools that we can use to predict reactions and enzymes. And then I will show you how we use these kind of tools to explore the biosynthesis pathway of a compound called noscapine. And in the end, I will also present you an online platform that people can use to do reaction and enzyme prediction that we also developed at EPFL. So most of you have probably seen this map of metabolic compounds and reactions from the KEGG database. So on this map, you see each dot represents a compound, and each connection between two dots represents a reaction. And this is basically this KEGG map represents our current knowledge of metabolism. However, we know that there are many things that we don't know about the chemistry that is happening in living cells. So probably our knowledge rather looks more something like this. And people have called this the dark matter of metabolism because there are many chemical processes that are happening in cells that we don't know about and that are very difficult to quantify. So we don't know how much and what we don't know. So here, now I want to give you a short overview on where these knowledge gaps in metabolism come from. So one part are unknown gene functions. So a lot of genes have been sequenced, but many of these open reading frames, we know that they code for proteins or enzymes, but we don't know what is the functions of these genes. For example, in E. coli, even nowadays, so E. coli is one of the most studied organisms, even nowadays there are like 25% of open reading frames without a functional annotation. Then we also have the case of orphan compounds. So these orphan compounds are molecules that are produced in nature, but we don't know how they are produced in nature. So we can measure them with mass spectrometry, but we don't know what are the synthesis pathways of these molecules. Then there are also metabolic interactions with synthetic molecules. So for example, drugs or pesticide or industrial chemicals, pollutants, they can end up in the environment where they're chemically transformed by microorganisms, but we don't know how they are transformed. And then there's also the case of promiscuous enzymes. So in this case, we can have enzymes that have different degrees of substrate specificity. So some enzymes, they're just very specific and only recognize one single substrate. But then there are other enzymes that can act on a broad range of substrates. And this is called enzyme promiscuity. And while we usually know, for example, the main substrate of an enzyme, it is possible that this enzyme also catalyzes alternative reactions, usually at a lower rate, so more difficult to detect. And yeah, usually these alternative activities, we don't know about them. And then we also have the case of orphan reactions. So these reactions, we know that they can happen in a cell, but we don't know which enzyme is catalyzing them. So what is the problem with these knowledge gaps? The problem is, first of all, they limit our basic understanding of the chemical processes in nature. And more specifically, for example, we have the scientists that will ask which are the reactions and enzymes that are responsible for the biosynthesis of an experimentally measured metabolite, or what are the biodegradation pathways of a given compound. And then the engineer would ask quite similar questions, which is which reactions and enzymes are needed to produce a target compound from a given substrate. So the engineering and the scientific aspects, the questions are formulated a bit differently, but in the end, it's the same thing, right? So the question is how we can generate hypotheses on possible biosynthesis pathways. And for this, we can use computational prediction tools. So yeah, what are the tools that we can use to predict enzymatic reactions? And it all starts with how we can do reaction prediction. And if we want to predict reactions, we actually need somehow to encode the biochemistry into the computer. So we can represent molecules as mathematical descriptions of the molecular structure. And then for the enzymes, we can use enzymatic reaction rules. And then if we apply these enzymatic reaction rules on these molecular structures, we should get the biochemical reaction description. Now, if we want to represent all metabolic reactions in nature, we need also reaction rules that represent all the known enzymes. Now, fortunately, the biochemical reactions have been categorized in only 6 classes. So there are only 6 basic types of chemistry that are happening in cells. Now, if we want to represent the 8,000 known enzymes, we figured out that around 500 reaction rules are sufficient to represent these known enzymes. So the number of reaction rules depends a little bit on the tool. Here I put 500 because several tools have around this number of reaction rules. But yeah, today I'm only going to talk about the tool that I used in my PhD thesis, which is called BNICE. And BNICE is actually one of the older tools. It has been originally created at Northwestern University before 2004. And then it has been further developed at EPFL in the past 10 years. It has been applied for 2 different case studies, in particular to biosynthesis and biodegradation problems, but usually more on the theoretical level, without experimental validation. Now, BNICE, what is particular about it is that the reaction rules that it has, they are manually designed and curated by biochemists, and they therefore basically represent the textbook knowledge of metabolism. I just very quickly mentioned that there are similar tools out there. Some of them, they also have manually curated reaction rules, for example, EnviPath and PathPred, which is the tool used in KEGG. And there's also RetroPath, for which there are several versions, and RetroPath relies on machine learning to deduce the reaction rules. So from now on, I'm only going to talk about how the reaction rules work in BNICE. So in BNICE, the rules are based on the Enzyme Commission, the EC classification system, which has 4 levels. So the first level describes the general type of reactions, and there are 6 of them. It's the 6 types of reactions that I showed in the previous slide. And then the second level describes the functional group of the reaction. The third level describes the cofactors that are involved. And the last level describes the substrate. So now the particularity is in the reaction rules, the substrate is not defined. So what does this mean? So let's say we have a reaction turning ethanol into the alcohol part and transform it into the aldehyde group. Then we can apply this rule also to other molecules that have the same reactive site and then perform the same reaction. And with this, actually, the reaction rules, they represent the substrate promiscuity of enzymes. So here, just to show how this looks like. So if we have a molecule and then we take a reaction rule and apply it, then the reaction rule will in the first step recognize the substrate at a specific, so the specific part of the molecule, and transform this part of the molecule into the product. And then we have different reaction rules that recognize different parts of the molecule and perform different reactions. And yeah. And then we can actually go one level further. For example, we can use these rules to generate a network. So when we take a source compound, we can apply all the reaction rules to the source compound, and we get a range of products. And then we can take these products and again apply the reaction rules to them and get a second generation of products, and so on. And with this, we can predict a network of reactions and associated products. So now this was probably all quite theoretical. And this is why I'm now going to show you the showcase of noscapine. In this project, we explore the biosynthesis pathway of this compound called noscapine. And yeah, we used B-Nice for that. So noscapine is a very interesting molecule. It's a secondary plant metabolite from opium poppy, and it is used as is marketed as an antitussive, so cough suppressor, and it can also there are also clinical trials for noscapine as an anti-tumor agent. Now, noscapine can be sourced from opium poppy, but the problem with agricultural production is that, for example, first of all, the noscapine content varies a lot in the plants depending on environmental factors. And then usually also the opium poppy doesn't produce a lot of noscapine, it produces more other compounds. And that means that you need a lot of opium poppy. And then you have the problem that this requires a lot of land, it doesn't grow everywhere. And you also have to guard opium poppy fields because people can get intoxicated from them. An alternative would be chemical synthesis. However, when you look at the structure, you see that it's very complex. And also chemical synthesis needs sometimes toxic catalysts, and you produce waste. So this is also not an ideal solution. And for these reasons, in the lab of Professor Christina Smoky, with whom I was collaborating, they created a yeast strain that can produce noscapine from sugar. So here, I show this is an abstraction of the noscapine pathway. And yeah. So they managed to create this yeast strain to produce noscapine. And then this was the part where I went to their lab and I started to work with them. And now one very interesting thing about this class of compounds, the class is called, oh, sorry guys, I think my PowerPoint just didn't make it.
No problem.
It's restarting. Okay, it's back. Almost. That's the thing that I was afraid of. I need to reshare my screen, I guess.
We can see your screen already.
Okay.
Yeah, we are there.
Perfect. Exactly. So this class of benzyl isoquinoline alkaloids, or BIAs, is very, very interesting because these plant natural products, they have very diverse pharmaceutical properties. For example, there is morphine, which is used as a pain medication, or papaverine, which is used as an antispasmodic drug, apomorphine, used as a treatment against Parkinson's disease, or even tubocurarine, which doesn't have any medical applications, but it's also known as arrow poison. So it has strong interaction with human metabolism. And our question was, what other compounds can be produced from this pathway by derivatizing noscapine pathway intermediates in the yeast? And so for this, we took our tool, Be Nice, and we applied now these reaction rules to the pathway. So here, we just applied it to this part of the pathway, starting from norcochlorine, which is the precursor of noscapine. This is what we got. So we applied the rules for 4 generations, and we generated this huge network around the noscapine pathway. And we found around 1,500 different benzyl isoquinoline alkaloids within this network. And then our question was now, which of them do we want to produce? And for this, we tried to rank these compounds by popularity. So we went to PubMed and PubChem, and we checked how many citations and how many patents were associated to these compounds. And then we found one that matched our criteria. So it was quite highly cited. It has 350 citations and 355 patents. And it's called tetrahydropalmitin. And it is just one step away from one of the intermediates in the pathway. So we thought, well, this looks like a good target compound to produce. So tetrahydropalmitin also is a quite interesting compound. It's traditionally used in Chinese herbal medicine, coming from different plant extracts. The plants that produce it naturally are some species from Corydalis and Stephania genus. And yeah, it's also currently on the market, and it's used as anxiolytic and analgesic to treat drug addiction from benzodiazepines and opiates. So we found that this was a good target, and then we tried to engineer yeast to produce this compound. Yeah, so here you have again the pathway to the first part of the pathway that we used. So here is norcochlorine, the precursor compound, and then here is tetrahydrocolumbamine, which is one of the intermediates in the pathway, and from there we then wanted to produce tetrahydropalmitin. Here you also, for those who really like metabolism, this norcochlorine actually comes from the shikimate pathway. Shikimate pathway produces dopamine and 4-HPAA, which are then put together to norcochlorine. And all these enzymes here involved until this precursor compound, they were taken from opium poppy, so Papaver somniferum. So we had this predicted step, and then we needed to find an enzyme that could catalyze the step. Unfortunately, in our lab, we also have this tool, Bridget, which has been developed in the past and which can predict enzymes for an enzyme for a novel reaction. So the basic assumption of Bridget is that structurally similar reactions, they can be catalyzed by the same enzymes through enzyme promiscuity. So here we have the predicted reaction, which is novel. So we don't know which enzyme can catalyze it. And you see here what is happening. You have O-methylation on this hydroxy group. Then Bridget, it calculates a reactive site molecular fingerprint. And it then compares this chemical fingerprint to a database of known reaction fingerprints. And then it can calculate a similarity score, and it can give you back the most similar known reaction that has an enzyme associated. And in this case, we found this enzyme here with EC number 2.1.118, which can turn columbamine into palmitin. And if you see here, the structures are very, very similar. It's just here in the gray zone, it's a little bit different, but other than that, the reaction is quite similar. So here's the query reaction that we wanted to engineer. And here is the enzyme with the top score that was predicted. And we found this enzyme in Coptis japonica, so one of the plants that also naturally produce tetrahydropalmitin. And then we put this enzyme in the yeast, and we saw that we can actually produce tetrahydropalmitin. So we were very happy. Now, however, when we didn't put the enzyme, we also observed that there was still production of tetrahydropalmitin. And the thing is that in the pathway here before, we have a lot of O-methyl transferases, and we suspected that probably one of them can also catalyze this reaction, but this hasn't been observed yet. And we finally figured out that this enzyme here had also a promiscuous activity and could produce very small amounts of tetrahydropalmitin. In the end, the titer was a lot lower, so we proceeded with keeping the enzyme that we found. Yes, so with this we achieved our goal. We had a new strain that could produce tetrahydropalmitin. And then, however, when we submitted the paper, the reviews were like, just one compound, that's not enough. And to prove the concept, we had to produce 3 other compounds, which we managed without bigger problems. So we also have strains now to produce armepavin, laudanine, and nandinine. And we used exactly the same procedure as we used for tetrahydropalmitin. So what did we learn from this experience? First of all, we learned that there's still a big potential for discovery of metabolite, reactions, enzyme, promiscuous activities in particular. What we also learned that this whole project took a lot of time, a lot of communication between the very technical part, so the very technical computational part at EPFL, and the experimental part at Stanford University. And it needs a lot of, you have to be very good at communicating if you try to work together between computational and experimental people, which is not always easy. And we figured out that we still managed to finally publish our paper in Nature Communications just a few weeks ago. And what we realized is that what is needed is a direct access for people who run the experiments to these computational resources. And this was, even though there are a lot of tools that exist, still it's very, very difficult if you're not a bioinformatician or a cheminformatician to understand and actually use these resources. And what we also learned is that, so this noscapine pathway is just a very, very small part of our initial KEGG map that we looked at. So if we now take one step back, and think about it. So we had 17 pathway intermediates, and from there we could predict 1,500 derivatives. And then only 6.5% of these derivatives were actually known to a biochemical database, so KEGG, for example, or similar databases. And the big majority of these molecules, they could only be found in PubChem, and they were not linked to any biological database. So, yeah, we thought that there's a big potential still to fill the knowledge gaps that are present in the rest of metabolism. And we wanted to find a way to make predictions for all of biochemistry at the same time. And yes, so with this, I come to this last part, Atlas X, the Atlas X project, where we try to develop a platform for biochemical predictions. So when we saw that there was really a need for large-scale predictions, we started with collecting all the biological molecules and reactions from publicly available databases, because now much of the data is distributed in different databases. So, for example, those who do more metabolic modeling, they have their own databases, and then there are databases just for drugs. And we try to compile all this data together in one database, and we found around 56,000 metabolic reactions, and also around 1.5 million molecules that had a biological or bioactive annotation. Now, out of this 1.5 million molecules, only 50,000 were actually involved in the metabolic reactions. So this is very few if you consider that all the biological molecules, they somehow have to be produced, right? So this also shows again the extent of the knowledge gaps. Yeah, so we took all these 1.5 million molecules and we applied our B-nice reaction rules for one generation. And what we got was a huge network of predicted reactions. So we predicted, starting with these 1.5 million molecules, we predicted 5.2 million reactions that involved 2 million compounds. So compared to what is currently available in metabolic databases, this is a lot. And then we thought that this would be a very helpful resource. So we created also an online platform, which is available at this URL here. It's called Atlas X Databases, and within now this huge network of predicted reactions, you can, for example, search for pathways between two compounds. For example, if you want to engineer a pathway from one metabolite to another, you can search for possible pathways. You can also do the same analysis that we did for noscapine, so expand an existing pathway, or you can explore around a metabolite of interest. What are the possible reactions that can happen around this metabolite? And also, very importantly, we have the enzyme prediction tool that is also integrated in this platform. So whenever you find a novel reaction, you can quickly check if there's maybe an enzyme that could do the job. Yeah, it's maybe not yet at the level of Google Maps, where you can look at it in a very super user-friendly way, but I think it's a first step into a good direction. So yeah, just to illustrate maybe a little bit how we can now use ATLAS-X to fill knowledge gaps, I took the example of the biosynthesis pathway of this compound here, staurosporine, which is a bacterial secondary metabolite produced by Streptomyces staurosporeus, and it has a lot of different biological activities. So yeah, quite interesting compound. And however, its biosynthesis pathway is very poorly characterized. So we know that it starts from tryptophan, and we know that the very last step, we know which enzyme is involved. But for everything in between, either we don't know the enzyme, so these are orphan reactions, or we don't even know the reaction mechanism that is involved. So yeah, we were wondering whether ATLAS-X can help us here. So we took the pathway and we expanded it for one generation. So we took all the molecules that were one generation away from each of these intermediates. And here what you see is, this is the tryptophan precursor. You see that tryptophan has a lot of derivatives. And here this is staurosporine, which also has quite some derivatives. And then, yeah, you can, if you do this, you can then go and check which are the possible molecules that can be derived from these intermediates. And then when we had a closer look, we figured out that actually for 5 out of the 6 poorly characterized steps, ATLAS can make a hypothesis. Try to characterize, for example, a biosynthesis pathway, it's already very, very helpful to just have a hypothesis about what can happen. So for example, here in the first step, we could propose an enzyme. In the second step, for this, we found a 2-step reaction from another database, actually. Or sometimes we found 3-step reactions, or we just found a potential enzyme. There was also one step where ATLAS-X was like, well, I don't know, which also means that it's not yet the final solution, but we need more efforts to figure out what is going on. Yes, so the ATLAS-X paper is currently on bioRxiv, if anyone's interested in having a look at that. Yeah, also a little bit more maybe about experimental validation that came from outside. For example, there was a paper that came out 1.5 years ago, where people used ATLAS reactions, so B-nice predicted reactions, to design novel 1-carbon assimilation pathways. So yeah, and in this paper, they could experimentally validate for predicted ATLAS reactions. And this is actually very important for us because at EPFL, we didn't do any experiments. So it was very difficult to say, guys, this is actually useful. But yeah, when other people actually have access to these tools, we now know that they can use it and they can work with it. And I also know that there are more papers to come, more experimental validations. But yeah, future will show how useful that is. So yeah, with this, I come to the take-home message of my talk. So yeah, prediction tools can generate hypotheses on biosynthesis pathway of different metabolites. These hypotheses are valuable for studying and engineering metabolic processes, and also very importantly, user-friendly platforms are important to make prediction tools broadly accessible. Yes, and with this, I would I like to thank my supervisors and co-workers at EPFL. First of all, Professor Vasili Hatzimanikatis from EPFL, who actually started the Be Nice tool many years ago. Then I also want to thank my close co-worker, Dr. Homa Mohammadi Pehani. She developed most of the database, and also she was very involved in the Bridget tool. And yeah, without her, this wouldn't have been possible. And also Anastasia Sveshnikova. She was also involved in the development of Atlas X. And then I would also like to thank my supervisor from Stanford University, Christina Smoky, and in particular also her postdoc, Dr. James Payne, who performed all the time-consuming experiments. And yeah, even in times of COVID, he managed to get to the lab and get this done. Yes, so yeah, thank you for your attention. And yeah, if you have any questions.